In this video, we're going to talk about glycolysis. And glycolysis occurs in the cytoplasm, in the cytosol. The goal of glycolysis is that we're going from glucose, which again is C6H12O6, and we're converting that glucose into two molecules of pyruvate, which is C3H3O3. And this happens using a series of steps. We're going to start with one molecule of glucose. Sometimes in my class, if you see me doing a shorthand for glucose, I'll just write GLU. The first thing that needs to happen is that it's going to get modified by two enzymes. We're not going to worry about the name for now, so I'll just name one enzyme 1, and the second one I'll say is enzyme 2. Again, these enzymes modify glucose, but to do that, one molecule of ATP is going to donate a phosphate to enzyme 1, and another molecule is going to donate a phosphate to enzyme 2. You can see I did a minus P with a circle around it. That means minus a phosphate group. So we go from adenosine triphosphate, tri meaning 3, losing a phosphate, to adenosine diphosphate, di meaning 2. When you lose two of these phosphates and you modify glucose with two enzymes, you get a different sugar, still with 6 carbons. And this sugar has the two phosphate groups that were donated to the enzyme. In order to make this first part happen, we have to spend, so minus, 2 molecules of ATP. The next thing that happens is that this new sugar gets modified by more enzymes. And we're not going to worry about the names for now. This time, instead of ATP, we have 2 molecules of NAD+ that are going to come in. Each molecule of NAD+ at those enzymes are going to gain an electron, which is my lowercase e with a little minus sign. And because we know that hydrogens have to travel with electrons, we're also going to say that they gain a hydrogen. So, when the NAD+ gains an electron and a hydrogen, you're left with a molecule of NADH. And because we have 2 molecules of NAD+, we get 2 molecules of NADH. This results in 2 molecules, and each of these are a 3-carbon sugar. I'd like you to notice here that we started with a sugar with 6 carbons, and our result was 2 times 3 carbon sugars. So we didn't lose any of our carbons. These 2 3-carbon sugars are modified by even more enzymes. This time, the enzymes are helped out by 4 ADP molecules. All 4 of these pick up a phosphate, and when adenosine diphosphate gets one more phosphate, the result is adenosine triphosphate. So 4 molecules of ADP each picked up 1 phosphate group. So what we get is 4 molecules of ATP. What also comes out of this process is some water, which, if we remember our equation for cellular respiration, you'll remember that water is an output. Most importantly, though, is what we get as a result of this modification. We will receive 2 molecules of pyruvate. For this to have happened, we gained 4 molecules of ATP. So I'm going to say plus 4 ATP. So the last thing we're going to talk about in glycolysis is the amount of ATP produced. So here's everything we have. And at this earlier stage, we lost 2 molecules of ATP. So I had -2 ATP. And then at this later stage, we gained 4 molecules of ATP. So for us to calculate the total ATP that was gained in this step, we have to add these up. And -2 plus 4 gives you +2. And we call that the net ATP. So the total ATP produced through glycolysis is 2.